Hello, everyone. Thank you for coming. It's very nice to be here. So as a kid growing up, I was really excited by Star Trek. Yes, I was a Trekkie. It's okay to say that, I think, and because I am a scientist. And I was always enamored by the idea that I could have all the tools and technology that I would need. And I would be on one of those starships, Enterprise or Voyager, and exploring the unknown. Going out there and seeking out new life and trying to make new scientific discoveries and going where no one else has gone before. That attitude and that way of thinking is what we do in our lab every day. And we are very interested in studying the brain and eye and how cells connect to one another, how cells grow and change over time, but also how they degenerate in conditions like Alzheimer's or glaucoma. So what you see on the right, you might think it's a planet in a faraway galaxy, or maybe a supernova, a bursting star, but it's not. This is actually 3D engineered brain tissue that we make in the lab. Now, this technology is not available on Starship Enterprise, but let me tell you, it's super exciting, and I'm going to tell you about how we use it. So how do we generate it? Well, it actually starts with a very simple vial of blood, the same vial of blood that you would give at a doctor's office or at a blood lab. We get that vial of blood, it comes to our lab, and we can extract out the blood cells. And then we add genetic factors to those cells, and then what you get over time in the lab are stem cells. So those blood cells reprogram into stem cells that divide, and we can make millions of them in the lab. We can change up the growing conditions once more, and you can generate three-dimensional tissue. And so what you see on the right is a brain organoid. So this is generated in 3D in the lab, but from a blood sample, a simple blood sample. Now this is revolutionizing the way that we do research now, because we can now have someone's brain in a dish, or at least some version of it. The cells that we have in these organoids, they form connections with one another, they're electrically active, and they form neural circuits similar to what we're finding in the human brain. So I'd like to share with you how we kind of use this in the lab to study brain disease. So on the left, what you see here is a brain organoid that's been engineered so that the brain cells are connected to one another, and it's the red cells connecting to the green cells. And this is generated from somebody who's healthy. And if we can do the same experiment from any disorder, so what you're seeing here on the right will be somebody who has a genetic form of epilepsy. And you don't need to be a scientist to see the difference here. There's many more cells and connections between them in this individual that has epilepsy versus the healthy condition. So what we're able to capture is something that might be happening in their brains, but in a dish, without ever having to touch their brains. So it's very powerful. And in addition, this is not only personalized, because this is coming from the single person that we're interested in, but also we can screen drugs and therapies in the dish first, before we go back to the patient. So we hope to be able to go from the right side back to the left side. In addition, at Krembil, we are absolutely enamored by technology, and we have such privileges here that we have access to some of the latest technologies released this year in 2024. It's a very fancy name. It's called spatial transcriptomics, but really what it is a very fancy way to genetically fingerprint each cell in that organoid. So why do we need to do that? So basically what you see on the left side is a very, very thin slice of tissue that's been put onto a chip. And it's that chip that has a genetic fingerprinting technology. On the right side is a blow-up. And you can see that each of those circles represents a brain cell, and the color represents a different genetic signature. So this is important for two different reasons. One is, it tells you what the cell is. So if you're studying Alzheimer's disease, you need to know which cells might be dying, but also the why. Why is that cell dying? We need to know the molecular fingerprint of that cell, because only then you know that, can you actually develop a therapy to reverse it. So as you can see at Krembil, science and technology is moving forward at warp speed. We have everything that we need here to be able to be leaders in the world in this technology. We have amazing basic science labs, and we work very closely with our clinical partners who give us blood samples, tissue samples, and sometimes even doing research in the OR. So this connection is very, very important. In addition, we have such a unique patient population here. And this patient population not only comes to the clinic for care, but they want to actively participate in research. This is absolutely necessary and very unique about Krembil. And finally, we have links to pharma and biotech. Sometimes you need some help to take a finding that's in your lab and to accelerate the development towards a therapeutic. So pharma and industry can do that. And in fact, this unique combination that you see here, this is the exact same reason that I moved my lab from a different institution in 2020. It was a very stressful time, but I wanted to be here at Krembil because I knew something special was happening and I wanted my lab to be a part of it. So as you can see, I'm super excited to be here at Krembil. I'm allowed to take risks every day in the research that I pursue, and I'm encouraged to do that so that I can do the science that no one else does, so that we can find cures to the different diseases that we study. Thank you very much.
So can I have some of those brain cells? Don't be selfish, right? You've got a lot of them.
We'll need your blood first.
That's okay. I'm ready to give it. This is amazing to me. I can barely get my head around it. So I read somewhere that we have 86 billion of these brain cells. Is that true? Around 86 billion?
Yeah.
And each one has...
Many more connections. But
okay,
yes.
okay, right. And each one has this unique fingerprint,
Correct.
Right, that has been identified. What is the potential now that you can identify and fingerprint all these cells? What are the implications of this for you?
So it's very big, and in fact, it's broader. So all the different disorders that you're going to hear about tonight, this technology, it's already here, and we're actually building around it at Krembil, and it's going to completely revolutionize how we study disease. We can do this for organoids, but we can also do this on primary tissue that we get from samples from individuals. So we can really understand the disease at the center of the disease. So that's the first thing that's going to be really exciting about this fingerprinting technology. The last thing that I'll say about is the idea that these organoids, they're not only good for studying disease in a dish, but we also think they're going to be useful for possibly replacing lost tissue. So in disorders where you lose tissue, and brain cells are dying, like in Alzheimer's or Parkinson's, these organoids might be able to be a medicine, a cell therapy for that person. And it's personalized because we're generating from them. So it's very exciting.
Amazing. Thank you so much, Karun. Thank you.